question. Can we bring back dinosaurs? If you think we can, please raise your hand. Alright, hold that thought.
My name is Andrew E.
My name's Katie Lamb.
I'm Ash Cavan.
I'm Lydia Montiel Villanueva.
Good evening. We all know that science has come a long way since the fossils were first discovered. We have cloned extinct animals, we have mapped genomes, and now some scientists believe we might one day bring back dinosaurs. But as the line between science fiction and science fact begins to blur, we are faced with a new dilemma. Just because we can, does that mean we should? Our literature review explores the possibility of resurrecting dinosaurs, their potential to survive in today's environment, and the ethical dilemma surrounding the extinction. First, what is the definition of a species? There are actually three definitions: biological, morphological, and phylogenetic. A biological species is a group of organisms that can interbreed and produce fertile offspring. An example would be where a poodle and a golden retriever interbreed and produce puppies that can interbreed on their own in the future. A morphological species is when a species is grouped together by its physical traits such as shape, size, and structure. Lastly is phylogenetic species, which is the smallest group sharing a common ancestor based on genetic and evolutionary history. For example, two birds with slight differences in DNA might be considered different species even though they look alike. So, if we were to bring back these animals, would they be considered as the original species or different, and how different?
Paleontologist Jack Horner and developmental biologist Dana Rashid are leading a project called Dino Chicken. Their goal is not to clone dinosaurs, but to bring back some of their physical traits by working with modern chickens. Chickens are close relatives of non-avian dinosaurs. Over millions of years, many of their ancient features have become inactive, but their genes are still there. Using a gene editing tool called CRISPR, the team is working to switch these genes back on. Cloning is not an option because DNA breaks down after 6.8 million years. Since dinosaurs went extinct around 66 million years ago, their DNA is too damaged to use. That's why this method of reverse engineering is a better path forward. Some chickens have already shown natural mutations that produce small traits like teeth. Even with physical changes, chickens still act like normal birds. While the Dino Chicken Project focuses on activating ancient traits in living birds, other projects are using genetic engineering to recreate extinct mammals more directly. The direwolf and the prairie ibex are the best examples of this.
Okay. The Pyrenean ibex was a wild mountain goat native to the high altitude regions of Pyrenees between Spain and France. It was most known for its large curved horns and rugged mountain lifestyle. It became extremely rare due to overhunting and habitat pressures. The last known Pyrenean ibex died in Odessa National Park in January 2000, marking the extinction of this subspecies. However, scientists have preserved its genetic material. In 2003, they used cloning technology to create a clone. The ibex was the first extinct animal ever brought back to life. The clone survived only a couple minutes, the exact time isn't known. It died to a lung defect, making this a failed clone, but it marked a major moment in biotechnology and conservation history. Next is the direwolves. Some people call it a success, but is it? On April 7th, earlier this year, Colossal Bioscience announced they brought back a direwolf, a species that went extinct 13,000 years ago, using ancient DNA from fossils up to 72,000 years old. They reconstructed the direwolf genome. Although direwolves and gray wolves look similar, they diverged from a common ancestor 4 to 5 million years ago and are genetically distinct. Researchers identified about 80 key genetic differences and used base editing to make 20 specific changes, targeting traits like body size, skull shape, and coat color. 2 pigment genes were disabled to give the animals white fur. However, studies have actually suggested that the fur wasn't white and instead gray. The edited cells were cloned and then implanted into dog surrogates. Out of 6 pregnancies, 3 pups were born, named Romulus, Remus, and Khaleesi's, and now they live in a secure reserve. However, many scientists are skeptical. They argue that 20 edits aren't enough to recreate a species that diverged millions of years ago. These animals, they say, are genetically modified gray wolves, not true direwolves. So if we could realistically bring back dinosaurs, would they be able to survive today?
To answer that question, we first need to know the difference in climate between the Mesozoic and current day. Temperatures during the Mesozoic range from 14 to 25 degrees Celsius. This higher temperature is due to the high CO2 levels, rising sea levels, solar luminosity, and changing geography. Also, something to note is that there were no large ice caps at the poles during this time. Climate today is actually much cooler and less stable than what we believe. The global temperatures today hover around 14.9 degrees Celsius, which is attributed to the ice caps of the poles. Also, CO2 concentration during the Mesozoic ranged from 1,000 to over 2,000 parts per million, compared to only 420 parts per million today. This could prove to be a serious challenge to dinosaur physiology as they relied on the higher temperatures of the Mesozoic to regulate their body temperature. Also, rapid seasonal fluctuations, colder winters, and greater climate extremes also rarely happen during the relatively stable greenhouse climate of the Mesozoic. Additionally, human-driven climate change is also a major issue. It is occurring far faster than any natural shifts in the age of dinosaurs, and it's something that they're not used to. Lastly, vegetation and atmospheric composition and the overall world that they used to live in make it highly unlikely they will survive in today's climate. Other factors to consider are food sources, stress, gut microbiome, and so many more. The one I would like to dive into is their gut microbiome. Most, if not all, animals live in symbiosis with the bacteria in their guts. These bacteria help break down and digest nutrients that we cannot replicate during any of the extinction process. So, if we were to rank these animals, would they become malnourished or starve to their death? So, after knowing all of this, and going back to the question I asked at the start, just because we can, does that mean we should?
Making dinosaurs isn't as simple and as plain as people say. There are three primary methods of the extinction: reverse engineering, genetic engineering, and cloning. Reverse engineering uses living animals related to extinct animals uses living animals related to the extinct animals, and scientists change the DNA to make new animals since genes can't be changed after birth. Both genetic engineering and cloning need viable DNA from the extinct species. Viable DNA or cell nuclei from extinct species. These genetic modification experiments require a large amount of test subjects to get a fully successful result. Test animals undergo various invasive procedures like vasectomies, surgical embryo transfers. An example is the Iberian ibex clone. Scientists used the DNA of Sealy. Sealy was the last Iberian ibex, and they implanted her DNA into 57 goats. Through this progress, only 7 were impregnated, and only 1 goat carried the pregnancy full term. One baby goat was born, and it survived around 7 to 10 minutes, and it died due to organ complications. If it takes multiple trials, is it truly beneficial? It raises concerns on animal rights and the exploitation of animals to get results. Nature has taken out these extinct animals already. Some scientists believe it would be non-beneficial to bring them back in the world, non-beneficial to them. The Earth is completely different now than before. If we brought back extinct species, they would most likely be placed in enclosures. Zoos are already controversial enough from taking an animal not being able to have the right environment for them. Being placed in isolation causes behavioral issues and makes them unsuitable for the wild. Then, if dinosaurs are going to be brought back, they could potentially be an invasive species having no competition and being in the place they don't know at all. That would explore and ruin ecosystems already in place, ruining the balance of our Earth now. We have enough species as it is, going extinct, and some have already been fully gone from this Earth, like the Tasmanian tiger, white rhino, dodo, and golden toad, and so many more. There are about 900 extinct species, most of which are 86% are animals, and the remaining 14 are flowering plants, except for a few fern and moss species. Shouldn't we focus on the animals we have now and not in the past? Is it truly beneficial to bring back dinosaurs when there are more consequences than benefits? Haven't really seen the consequences by watching Jurassic Park. It might not be exactly accurate, but it does represent the dangers and moral concerns with messing with concepts today humans can't fully understand. Are we reviving the past or risking our future? Dinosaurs were alive 66 million years ago. Our animals today are already slowly going away due to human impact, pollution, climate change, and much more. Animals have successfully been brought back but barely live for a couple minutes or live under extreme limitations. These efforts show that extinction is not a perfect fix. It's a glimpse of what we already lost. It takes many procedures or doesn't always go as planned. Shouldn't we relocate those resources to something else, something better? Is it beneficial, or is it just for entertainment to see how far we can go? Our sources. Thank you so much, and thank you to our advisors, Kayla, Juan, Megan, and our fellow Cameron. Thank you to our Laura Sam, and thank you to Meredith College and our staff. Any questions? You mean for the Iberian ibex, why do they use goats?
Like, why do they choose goats?
They use genetic engineering, which uses. Oh, sorry. Why do they use goats for the Iberian ibex? For the Iberian ibex, I believe they use. Sorry, yeah.
The reason they use like goats is that to clone back animals, we need to use their closest relative in order to actually clone them, and the closest relative to the Iberian ibex was the goat.
Okay, thank you.
What's each of your favorite dinosaur?
Favorite dinosaur?
Yeah.
Mine is a Stegosaurus.
Mine is the Dilophosaurus.
I don't really have one, but I guess I say the T-Rex.
Mine is the Iguanosaurus.
Cool.
Good afternoon, everyone. My name is Uzma.
My name is Natalie. I'm Laurel. And I'm McKinley.
And today we're presenting our experiment on tampons and which brand is safest for vaginal pH. Previous research from UC Berkeley has found that toxic metals such as lead and arsenic has been found in tampons. These metals are easily absorbable to the vagina due to the stratified squamous epithelial tissue found there. We decided to explore the tampon's effect on vaginal pH since there was not much research done on that subject. pH is incredibly important for the vaginal microbiome to clean itself, so we gravitated towards this topic to research.
The question we were aiming to answer was what brand of tampons are least disruptive to the vaginal pH, and does the brand being organic play a role?
Based off of our question, we developed our hypothesis. If the pH of non-organic and organic tampon brands are tested, then the non-organic brands will have more alkaline pHs.
Before we got into experimental design, we needed to figure out what was already known about vaginal pH and why it was important. So for those that are reproductive age, that means after your first period, before menopause, the average pH is usually around 3.8 to 4.5, sometimes 5, which is very, acidic. It's more acidic than you would think. It's moderately acidic. The reason for this is because of a bacteria in the vaginal microbiome called lactobacillus. It processes glycogen to produce lactic acid, and that's what causes the pH. And the pH is really important because the acidity defends against infections like bacterial vaginosis and certain STIs. We also found that menstrual blood is much more alkaline at 7.4, which is important because if you're also using an alkaline tampon, then that difference in pH will be trapped against the vaginal wall and can potentially cause disruptions.
Alright, so how did we even come up with this idea? Because I know it's a little bit out there. But when we were thinking of this project, we wanted to combine all of our interests and it ended up resulting in reproductive health care. So after that, we wanted to test something that a lot of people who menstruate use, tampons. So we wanted to do this as ethically but also realistically as possible, which is why we decided to use vinegar in our water solutions for the beakers. So then after that, we went ahead and got our tampons, which are actually from our GSE community.
Thank you guys for donating. That was really helpful.
So yeah.
The materials we used for this experiment were 15 beakers, 3 beakers each for each of the 5 brands. We got pH strips from one of our advisors, Kayla. We used tampons, and the brands that we used were Tampax, Orchid, Vita, Honeypot, and Antflow, in which Honeypot and Antflow were the only advertised organic brands. We used distilled water, white vinegar, a pipette, tape, and a graduated cylinder.
So for our experiment procedure, we collected all the materials that Uzma just talked about. Then we went ahead and labeled all of our beakers with the respective tampon name, sample number, and if they had white vinegar in it. Then for our sample 1, we only put 100 milliliters of distilled water in there just to use it as a control. After that, for sample 2 through 3, we went ahead and added an additional 0.25 milliliters of white vinegar. And then after that, we tested the pH of each solution before submerging the tampons. We went ahead and submerged the tampons in there for 6 hours. We checked the pH strips. We checked the pH with pH strips. And then we went ahead and recorded this data. We went ahead and let them soak for another additional 18 hours so that we could get a total of 24 hours of them soaking and recorded the data that Paul was about to talk about.
So these are our lovely graphs. The timestamps are labeled. Pink is going to be the first pH measurement at 0 hours. Purple is 6 hours and blue is 24 hours. So the first brand, Orchid, experienced a slight decrease in pH, but it still stayed in the healthy range. And then Tampax, we saw very little change. There was a little bump up. That could have been due to an error in our measurements or just because we were using pH strips, or it could have been external factors such as the beakers being left uncovered. Next, these are our organic brands, Honey Pot and Aunt Flo. Aunt Flo is the first one. It also stayed roughly the same. There was a little dip. Again, that could be due to an error in measurement, but it was not like significant enough to change our results really. And then Honey Pot stayed completely level the entire time, no change in pH. Lastly, we have Vita. Vita got more acidic in our first sample and then more basic in another sample. That could be due to an error in measurement or just the beakers being left uncovered. We're not really sure why. In all, none of the brands had such a great increase or decrease to be outside of the normal vaginal pH range. And there was also no consistent differences between the organic and non-organic brands.
Throughout our experiment, there were a few instances where our results could have been altered or influenced by outside factors. For example, instead of using a pH sensor like we had originally planned, we ended up using pH strips. This was due to time constraints and also some technological difficulties. However, if we had used the pH sensor, we would have been able to get more exact numbers, including decimals for the pH, which we just weren't able to obtain using the pH strips. Some other examples of some errors we could have had included uncovered beakers, one tampon being sized super rather than small like the rest of them, and then also using different size beakers for each of the soakings. All of these things could have altered our results. To conclude, our hypothesis wasn't supported from this experiment. We discovered that a majority of the tampons experienced little to no change. While we did try to come up with a realistic experiment, we acknowledge that there's a lot we could have done differently if we weren't limited by time. And we would love to continue doing more outside research on this topic, specifically involving the pH effects and chemicals and metals included in tampons. Even though tampons have been around for a while, there's not a lot of research done on the pH from what we have found. This research is highly impactful because it affects many, and we should be more invested in questioning what we put in our bodies and if it is providing us with health and support. These are some of the references we utilize, and thank you. Are there any questions?
No questions at all? Yes.
I have a quick question. How do you think, like, I know you guys specifically looked at tampons, but did any of your research come up anything about like pads and if the different pH of those would also affect it in a different way?
So there is this thing called like pH control gel, and they sell it for tampons and pads. We didn't look too much into that just because it was hard to find and it was like just sketchy. We didn't know how we would test that even. But pads, if like they're kind of made of the same material, so they wouldn't have too much of a difference, and also they're not like being inserted, like they won't touch the vaginal wall, so it wouldn't cause a great pH shift. Just use them the right way.
Yes, it's your question.
Of the brands that you tested, would you like recommend any of them, even though I know the results were different?
I think like all of the brands, it really just depends on like what you personally prefer, because I don't prefer tampons, but I think the organic brands did show a lot of promise, and mostly when you look up like the lead and the chemical testing, they mainly did it on the non-organic brands, while the organic brands tend to have like less of those. So if you are gravitating towards a certain brand, I think Honey Pot had a great like advantage.
As someone that uses tampons, I hate Honey Pot, but that's just me. But do your research. I like Tampax, but it's up to you. Do your own research.
What's your question? So it had nothing in it. It's just a straight like vinegar or water, just water.
Any more questions or meeting? Okay, thank you guys. Hi. So our project was the effect of urban pollutants on lentil plant growth. My name's Lainey.
I'm Christina Polanese.
I'm Tessa.
And I'm Trisha. So, what is urban pollution? Urban pollution is the introduction of harmful substances from urban environments.
Why do we care? We care about urban pollution because it doesn't just affect air, it harms soil and plants. Pollutants from common household items prevent seeds from germinating and weakens plant growth. Understanding these effects help us make better environmental choices and protect our ecosystems.
So the question that we sought to answer is, how does urban pollutants affect plant germination and growth? The hypothesis that we formulated is if pollutants are added, then lentil seeds treated with bone and blood meal and lemon juice will grow more than the control. However, those treated with detergent pods, baking soda, dish soap, epoxy resin, and microplastics will grow less than the control. Our first experimental group is bone and blood meal. Bone meal is heavy in phosphates and blood meal is heavy in nitrates. If the effects that this has on plant growth is that it helps keep the plant's lush and green and helps support new root growth. But if it's in a heavy concentration, then it'll lead to plant death and roots burn. The industrial sources of this group is factories that manufacture fertilizers, wastewater treatment plants, and agricultural runoff.
Now, next we tested pH. We used lemon juice, which has a pH between 2 and 3, making it highly acidic, and then we used baking soda, which has a pH of 8.5, making it basic. Some sources that can affect pH are runoff from road salts and also from landfills, and then acid rain, which is from vehicle emissions and factories whenever they react with the water in the atmosphere. Some of the impacts is a low availability of nutrients to the plants. Some plants do like a slightly acidic soil, as it helps make the iron more soluble and available to the plants. But in both cases, aluminum becomes more soluble, which is highly toxic to plants.
And other items that we tested were dish soaps and detergent pods, which are like common cleaners that we all use today, but it is harmful to soil and plant life. They contain surfactants, which are like chemicals that break down oils. This can strip away the protecting coating on plant leaves and block water from entering the soil. In addition, pods are also concerning because they contain PVA, which is a plastic film that doesn't biodegrade easily. This can disrupt soil microbes, which are really essential for healthy plant growth.
Okay, so our last experimental group is microplastics. So to simulate this, we basically chopped up plastics as finely as we could. And then the other group we did was epoxy resin, which is a liquid plastic that hardens. So I know we all hear about microplastics in our bodies, just kind of everywhere, because they are everywhere. But what do they actually do in plants? So they clog the pores of the seed while the seeds are germinating, which causes water and nutrients to be unable to reach the seed. And then once the plants are actually in soil, they will kill the microbes that are in the soil that the plant needs to process nutrients. And then they come from littering and also mismanagement of trash.
So we did this experiment in two methods. First is the Ziploc method, and the other one is soil and cups method. To start off, for our Ziploc germination test, we basically took paper towels and soaked them in a 9-to-1 mix of water and pollution. Then we placed 7 lentil seeds on each of the saturated towels and sealed them in a Ziploc bags. These were placed under partial sunlight for 7 days.
So our next method was the soil and cup, where we used 45 milliliters of our soil and then 5 milliliters of our pollutant. We mixed that up, and then we used 3 seeds that were already germinated and placed them about an inch deep in the cup. We then left them in partial sunlight for 7 days, and we watered them with 2 milliliters of water every 3 days. And our control just contained water, no pollutants.
To reiterate, this is a snapshot of how the soils were arranged into these 9 cups, and the next slide shows that both the Ziploc bags and the soil cups were placed at the same location under partial sunlight. Next is our results, and we'll start with Ziploc. This is a picture of day 1, and you can see some color changes for a few of them, such as dark brown. After 7 days, we saw clear differences. The final one. So to start off, we can see in the control state, they show healthy germination, but for the lemon juice, they also showed some of the healthy germination as well, but not in the same size as the control did. Pollutants like epoxy resin, detergent pods, and dish soaps produced reduced growth, and they had color changes such as dark brown, as you can see for the baking soda as well. In addition to that, bone meal and blood meal also showed less growth, and even a mold has formed in the blood meal. Interestingly, the plastic group also had about the same results just as the control did. Next up, we have soil cups, and this is the layout of how the cups were arranged according to their pollutant, where each of them were placed with 3 germinated seeds. After 7 days, we took out the plants or the seeds and saw many differences. To start off, in the soil test, the control plants grew the best, but surprisingly, the lemon juice also had about the same results just as the control did, in addition to the plastics, which was also similar. In addition to that, baking soda had also a reddish color and bone meal had a black color, whereas the epoxy resin, detergent pods, dish soaps, and blood meal also had some sort of a color change, such as a tint of brown, but they all had little to no growth.
Okay, so here are our numerical results. So we basically averaged the different seeds or sprouts, the length of all the sprouts. So to start off with the control, it had pretty much the best growth, if you would say, overall, which makes sense since there was no pollutant in it. The plastic actually had a really, really good length in germination, which we'll get into why that is in a second. But the average length in soil was a little bit lower because one of them didn't germinate. But the other ones grew pretty tall. Epoxy pretty much cut off all the nutrients. There was pretty much no growth there. The bone meal and blood meal also were pretty stunted. They really didn't grow very much. And then the lemon juice had really, really good growth, which is our acidic pH simulation. And the baking soda, dish soap, and detergent all had little to no growth. So if you're a visual learner like me, I like looking at these graphs, which I think put it into perspective a little better. So we've got our control, our chopped up plastic, epoxy, bone meal, blood meal, lemon juice, baking soda, dish soap, and detergent. And then it is the same on this slide, but for the germination, that last one was for the soil growing. So,
Okay, so what does all of this mean? So basically, the bone meal and blood meal ended up having too many nutrients, which limited the growth of the plants. And next, for the lemon juice and baking soda, we learned lentils actually preferred the slightly acidic soil from the lemon juice, which caused them to grow very well. Whereas the baking soda was way too basic, ended up just destroying the plants. Now, for dish soap and detergent pods, the chemicals stripped the leaves of their cuticles, leading to decreased growth. And then for microplastics, the epoxy ended up hardening, so the plants were unable to get any nutrients. But what we found very interesting was our big pieces of plastic actually led to more growth due to the roots now having a structure to grow onto.
So, was our hypothesis supported? Partially. In regards to lemon juice, baking soda, dish soap, water, and detergent pods, and epoxy resin. However, those treated with bone and blood meal and plastic were not. And moving forward, we could conduct this experiment using different types of plastic, a variety of plants, different concentrations of pollutants, and testing soils of different natural pHs.
So here are all of our sources. We've got 2 slides for those. And thank you so much. We also wanted to thank Kayla for being a wonderful advisor, and also Juan and Megan and our wonderful fellow Cameron and everyone in Natty Sci. Y'all are awesome. So any questions? Saw your hand first.
Why did you use lentils?
Lentils are really fast germinators. They germinate really fast. So we had a limited timeline on this experiment, a couple weeks from the time that we actually kind of got our idea and everything together. So we wanted to make sure that they would actually give us results.
Also, we originally started with pinto beans and we saw zero growth whatsoever. We don't know why, they just did not germinate. So we were like, we don't have time for this, we're using the lentils.
Any other questions?
Were there any big obstacles during this project?
I'd say being consistent, like we forgot to take the pictures like of day 0, and also the pinto beans, like we originally were gonna do pinto beans and it just didn't germinate. So we had to switch to lentils, which was, both of those were in our like head, but yeah.
I think for the photos were so hard. They took a long time to upload into my computer. Well, that pissed me off, but other than that, everything was good.
And I would also say the microplastics, because realistically, microplastics are smaller than like a grain of rice, and we were trying to cut them down so teeny tiny. Tessa and Christina, they did a great job.
Yeah, I was also gonna say the microplastics, they're just not really microplastics. Microplastics are just, so tiny, but we did our best to try and simulate that. Are there any other questions? Yes. That's a good question, actually. We did not think about that. We would have to do some research into that. I think it would probably depend on the type of glitter, but I'm not a glitter expert, so I don't know. Other questions? Yes.
What inspired you to choose your research on this topic?
Inspiration?
Well, I like to say that I like to garden. My dad is a big gardener. We have like a garden in our backyard. But also we started off we kind of said that we liked chemistry. I know you did. And then we kind of shifted. I don't remember what we originally.
It was like most of the groups were doing testing on water, and we initially planned on doing water testing and all that stuff. But then since others were doing them, we wanted to do a change. So yeah.
Other questions? Anyone else? Well, thank you guys so much. You're gonna be amazing.
Alright, good evening everyone. My name is Braden Barnett.
My name is Ashton Kirkwood.
I'm Ovitel.
I'm Tommy Crowley. And we're so glad you're here this evening, and we're so excited to present to you all our experiment. As you can see, it's titled Mass Protection or Mask Hysteria. We'll get right into it. So to start with, I want to define what our question we were trying to answer through our experiment was. And this question is, what type of mask is most effective at stopping the spread of bacteria during coughing? And then you can see some of our materials on the right side of the screen, but I don't want to spoil anything yet, so I'm going to go ahead and pass it on over.
So why did we choose masks for experiment? Masks are one of the most common forms of PPE with uses in surgery, biology, and just general safety. But even since before the COVID-19 pandemic in 2020, a lot of people have questioned the safety of some masks, like the KN95 mask and the filter mask, over others, like the cloth mask. And so in this experiment, we just wanted to verify the claimed safety that those masks have.
So before we get into our experiment, we wanted to do some prior research on the topic. So looking, we found that yes, there was in fact differences in particulate penetration of different types of masks. So we see here with the 2-layer fabric masks tend to perform the worst. So the less layers you have, obviously the worse your penetration is going to be, or the better your penetration is going to be, because you have less chances to catch the particulates. Then we see the one with the best at 2.28% penetration was KN95. That's because KN95 masks are kept to a higher standard. They have to filter out at least 95% of particulates to be able to be sold and labeled that way. So that is a trusted mask type, so we made that one of our controls. We also saw that cloth materials in general had the highest particulate penetration. So we wanted to test cloth and paper to see how they perform against control.
Now that after we did our research, we were able to pose our hypothesis. Word for word, we state if low-quality cotton and cloth face masks are ineffective at blocking airborne pathogens, then agar plates exposed to coughs through these masks will show bacterial growth more similar to plates with no masks than to those with a KN95 mask. In simpler terms, we were saying that low-quality masks and cotton masks will be more similar to that of a cough with no mask on than that of a KN95 mask.
So some of the materials we used obviously include our 3 mask types, which we all got from Amazon, our 3-ply disposable filter mask, our 2-ply cloth mask, and our KN95 masks. And we also borrowed agar plates and an incubator from our instructor, Kayla Palia. And we also used gloves and tape just to prevent contamination for our agar plates.
Next step was to design our actual research. First thing we decided to do was prepare our plates, then labeling them. We had 9 different trials. Our first one, step 2, our atmospheric plate. All we did for this step was sit an agar plate out for 45 seconds and then cover back up. Our next step was to actually perform our coughing trials. We had 2 separate subjects perform this trial. The trial had our subjects wearing either no mask, a cloth mask, a throwaway mask, and then a KN95 mask. And then after that, we were able to seal them up and then put them in our incubator.
So once they were in our incubator, we would check them every day for about 1.5 weeks just to count how many colonies there are and also track them by photographing them. And you'll see those photos later.
See those photos now. After our first day, we only had a little bit of growth on one of our subjects, no mask cough. You can see, thank you for the pointer. On the bottom part of the agar plate, we have a little bit of growth. Then on our third day, we get some more growth on our no masks, and on our cloth, we finally get a little bit of on our same subject's cloth mask cough. And then for our 5th day, was when we ended it. That growth on the cloth mask grew a little bit bigger. And then on our no mask, those bacterial growths got bigger too. But other than that, there was no significant growth.
So after we counted up all of our agar plates, we found that there were 14 bacterial colonies on one of our no mask tests and 1 colony on one of our cloth mask tests. And so this shows that all 3 masks were effective at blocking bacteria, but the KN95 and the filter mask were more effective than the cloth mask. It's also important to note that we only found these results for one of our 2 test subjects.
So based on our results, we found that this experiment rejected our hypothesis. So these masks actually performed quite well. We found that all mask types reduced bacterial growth post-cough compared to an uncovered cough. However, we did see that the cloth mask performed slightly worse than the paper mask. So even though they're both effective, we did find that the paper mask was better than the cloth.
Alright, and as with any experiment, there's going to be some kind of challenges or potential errors we ran into. One of the biggest ones was that last one right there, the leakage from the side of the mask was not accounted for because we were coughing directly on the agar plates and testing for filtration, not necessarily how much bacteria was reaching the atmosphere. So that was probably one of the biggest potential errors that came along with our experiment. Also, some other things like coughs between subjects being inconsistent, that's the same for everyone. Even the same person might not have the same cough in every trial. Limited sample size, as we said, we only had 2 different subjects, so if we get more subjects, that gives us more consistent results. Airborne bacteria could affect plates in various ways that we don't even know about. It could have made its way into 2 of the plates and not the other 6, so we really don't know. Also, handling plates after incubation could introduce more bacteria. We made sure to wear gloves and reseal them every time, but still there's always a possibility. And then for the human error side of it, counting colony numbers, humans aren't perfect, and we didn't really have a perfect way to count the colony numbers for each of our agar plates.
So, in conclusion, we found a couple things. One, coughs were highly inconsistent, not only among individuals, but among coughs of the same individual. We saw that cloth masks were the least effective at stopping bacterial spread. We found that face masks were effective at stopping spread altogether, even though one may be slightly better than the other. We also found that the study only accounts for one scenario, so basically bacteria coming from outside, or from inside to outside. This doesn't look at the scope of if it's going to protect you from outside to inside.
And I want to take a moment to say thank you to all of our instructors for their guidance and providing the materials for us. And now I want to open the floor for any questions. that was a controversial topic of whether we would say it or not, but I think it's fine to say it was Tommy and Ashton were our two subjects that did the call.
Who was cleaner?
Tommy said less break.
Any other questions? Right here.
When you were talking about the how many times did you call?
So we tried to limit it to 3 calls and try to make them all the same, like, consistency. So the same amount and the same period of time. Obviously, like I said, there's a very wide margin of error in that, but we tried to make it as consistent as possible. Right.
I know you tested, like, the way you tested is basically if a person was wearing a mask who was sick. Do you know how you would test it if you wanted to test, like, if someone without a mask coughed and then how much bacteria would get in if someone else was wearing a mask? Do you need
So are you asking if like 2 people are wearing a mask?
yeah, that or like if the person who was coughing wasn't wearing a mask and then like I'm sitting there with a mask on and then how likely is the bacteria to be transferred or virus bacteria whatever to be transferred?
Yeah, so like Ashton said, we more measured about like bacteria going from inside your mouth to outside in the agar plate. But I guess if we would repeat this experiment, we could just like place the mask on top of our agar plate to test like from your mouth, like bacteria going from your mouth to on top of the mask and see if it reaches in the agar plate.
Thank you.
Do you know how your results might be different if you were testing for a viral infection versus the bacteria?
So yeah, we used agar plates for this, and agar plates are good for like measuring bacterial growth. And yeah, we couldn't really measure the viral growth, but there's probably some way you can test it.
Most viruses do travel through like saliva particles, so not really like floating through the air, but during a cough, they're attached to the particles. And this more so measured those spit particulates during a cough. So I think they're slightly linked, but it doesn't account for more airborne pathogens.
Okay, thank you.
Good afternoon, GSE friends, family, and staff, and welcome to the happiest day at Gov School East. Thank you for coming to our Natural Science Cornerstone presentation. I'm Ian Kim.
I'm Nora Faustino. I'm Emma Walters. Yes, you are.
And I'm Joshua Morrell.
Before we dive into our experiment, by a show of hands, who here has ever used a water fountain? Oh, everyone. Over the past couple weeks, we aimed to find out how nasty the most disgusting and unhygienic object at Meredith College was. No, not the moldy bathrooms of String 2, or the musty halls of Brewer, not even the germ-covered handles of the dining hall doors. Today, we will be talking about something you may have even used before coming into this presentation, the water fountains. We really hope you didn't use them.
From the start, we wanted to find the grossest thing at Governor School East and see how much bacteria is on said thing. Initially, we wanted to look at hand dryers in bathrooms, but ultimately we realized there are no dryers on Meredith College. So after some brainstorming, we landed on our second best option, water fountains.
We want our experiment to be based on something that was frequently used by a wide range of people, and I don't think I can name a better thing than a device people slobber all over all day.
First, we asked the question, which of the water fountains at Governor School East has the most bacteria growth? Based on that, we formulated a hypothesis that states, if we test 7 water fountains at Governor School East, then the gym water fountain will have the highest count of bacteria when cultured in a petri dish because people are more likely to need water in an athletic facility. With our experimental procedure, we applied prior knowledge, research, and advisor assistance. For our preparation, we ordered 10 pre-poured agar petri dishes and swabs. Then, to collect our samples, we selected a few fountains around the school and gathered data by taking our swabs and thoroughly circling each dish
each swab around the spout of the water fountain. For our control group, we did the same exact thing, but first we cleaned it with Dawn dish soap. For our procedure, we transferred the bacteria onto agar plates by rubbing the swab in a zigzag motion across the entire dish, labeling each one after with its corresponding water fountain. On the initial day, we placed each plate upside down in the incubator. Each day that followed, we checked on the plates by taking pictures and avoided letting them get dried out. If they seemed to be dried out, then we added 1 milliliter of distilled water to each and placed them upright in the incubator. After 7 days, our results were shown by counting the amount of bacteria colonies on each. This helped us determine which water fountain had the most bacteria and which fountain had the most colonies.
Alright, now as you can see, on the right we have our independent variable, which is we use different water fountains for this experiment. And on the left, we have our dependent variable, which are the number of bacterial colonies used that we found on the petri dishes. Now we're going to show you guys our control for this experiment. So as you can see here, this is from the science building. It's not that dirty. And then we have, and then this is our actual one, which is still not that dirty, but it's just been, there's like cracks in like the petri dish, but overall it's not that dirty. So now, does anyone live in Brewer? All right, well I advise you guys to look away. Yeah, to be honest, actually wasn't really that dirty compared to the other ones that we have. And what about Van? Oh wait, that's right, no one lives in Van. Oh, you do? Okay. Okay. And then this one, it has large spots of colonies, especially on the 4th and the 8th. But overall, both of these were touched the least out of all the ones that we've done.
Alright, so next we have Poteet. Where are the Poteet girls at? Yeah! Y'all might want to look. Because these are Poteet water fountain samples. Yeah, not the prettiest. And after Poteet we have String. String? Anyone? String? Y'all might also want to look away, because these are the String water fountain samples. Not looking good.
Now, is anyone Area 1 choral music? Woo! Those are your water fountains. Next, does anyone go to open gym every day at 4:15? This is what your water looks like. So each one had similarities with their shape of irregular or circular forms. We didn't have time to examine under a microscope, but we can hypothesize that these bacteria could be forms of Listeria, which could be even novi, or 2 different types of Salmonella, which are Tecumseh and Shigella.
So, y'all ready for the top 2 grossest water fountains at Meredith College? Yeah. Can I get a drum? And we have Jones Chapel and Weatherspoon Gym. These are the ones with the most bacteria after 4 days. Jones Chapel, you maybe already see, was mainly because of the tiny colonies condensed on the left side. They were yellow and there were a lot of them. And then Weatherspoon was in a similar situation, but they were more spread out throughout the whole entire agar plate. After 7 days, we have the same 2 colonies, except the amount of colonies decreased, which can be attributed to colonies dying or merging together.
So here is the chart of all of the colonies that we had to count, and as you can see, both the science building and Van probably had like the least amount of colonies. Poteet and Spring were steady throughout, but the chapel and gym had like the highest number, and it was a pain to count. Right, Eden?
Yeah. So our biggest obstacle was just with our petri dish care and handling. We saw a lot of agrodesiccation, which is essentially just the drying out of the agriculture medium. This inhibited our bacterial growth by applying a limiting factor to one of the 3 essential things for growth, which is moisture, heat, and nutrients. You can see the desiccation or the drying out in some of our previous petri dishes through the contraction of the agar inside the dish or the cracks that you would see before. It also made it really hard to count some of our colonies on our later days, like day 7, right, Ian?
I'm so tired of counting colonies!
Okay, so this is where we saw the most desiccation, our original control. It's pretty bad. But we ended up having to redo it because we could not salvage it, so on July 4th, we decided to redo our control group, which extended our experiment by 3 days, but we knew it was really, really critical to find our data and analyze our results. It also helped us adjust our procedure to preserve our other petri dishes, which were facing minor desiccation, by adding 1 milliliter of distilled water every day that it looked like they were drying out, which was most days. So in conclusion, our results did not support our initial hypothesis. Our results supported the fact that the Jones Chapel had the most bacteria colonies after both 4 and 7 days. We hypothesized that this is because the Jones Chapel is not only used by Meredith College, but is also used for other public events such as weddings or just other events. This is our resources. So by a raise of hands, who is going to be using a water fountain around Meredith College after this? Okay, more than you than we thought. Any questions?
No.
Yes.
How many water fountains are on Meredith?
The question was how many water fountains are on Meredith, and I don't think we know. But we'll look into it.
Some of the water fountains have like the bottle dispensers. Did you think to test any of those, or?
So the question was that some of the water fountains have bottle dispensers. Did we test any of those? And we didn't test any of those because not all of the buildings that we decided to test had them. So we wanted to keep it consistent with the same spout that you may put your mouth on if you were to get water straight out of it. You also fill it up with your water bottle if you need to.
What part of the fountain did you swap?
Yeah, so the question was, where did we swab around the water fountain? We swabbed around the entire spout. So where the water comes out, and then around the back of the swab, the spout. So any water that could potentially splash when you're getting water, or if someone breathes on the spout, that's where we swabbed. Any other? Yeah.
Why did you not test Faircloth?
Why did we not test Faircloth? We only had the ability to test a couple different water fountains. We only got 10 different petri dishes, so we didn't want to test too many, also to present in this given time.
Time limit.
Yeah.
Faircloth does not have water.
Oh, okay, perfect. There you go. Yeah.
Is there a legal level of bacteria that you're trying to
Is there a level where it becomes safe or not?
Yeah.
Yeah. We did not research that, but that's honestly a really good question. I think that would be really interesting to look at. I'm sure people are aware of the amount of bacteria on water fountains.
And we also don't account for janitorial staff cleaning the water fountains or anything like that in this test. So we don't know their schedule or anything like that. So no offense to the janitorial staff. I'm sure they do an amazing job, and we love them here at Meredith College.
Yes, we do.
Any more questions?
I just want to say, could this give credence to instructions of not putting the water bottles up to the common ice dispenser, water dispenser, but rather filling with a clean cup? What do y'all think?
So we think that this probably does really correlate because a used water bottle, that's going to transfer bacteria in the agar dishes, like it is able to grow. But also the ones that we tested, we didn't test the ones in the cafeteria and stuff. Most of the time, you're probably not filling it up with your water bottle and you're probably getting it directly. So we wanted to also account for like straight saliva and stuff. But we do think that there'd probably be a decrease if you were to use clean cups.
Yay for clean cups.
Yay!
Any more questions? Thank you.
Thank you so much for being with us tonight. We are one station of our natural science cornerstones. Our students have been working really hard over the last 3 weeks to either conduct an experiment or a literature review on the topic of their choice. For today, we do encourage you to record your own students in their group. We will also be recording in the back, so these will be posted later. We ask that you try to stay here during the presentation, and then we will give 3 minutes of rotation time. So especially students, if you're trying to make it to another rotation, you will absolutely be able to. Students, just a quick reminder, at 10 minutes into your presentation, I'll give you a 10, and then you have 3 minutes remaining to finish your presentation and answer any questions. For the audience, we will have questions, an opportunity for you to ask questions. We encourage you to ask them. Just remember, our students have worked really hard. We're excited to share this information with you. So our first presentation, and we'll start right at 4:15, is the impact of food dyes on oral bacteria. So I will start my timer. Very well last minute. Thank you for being here for your students.
Good afternoon, everyone. My name is Steve Yoker.
My name is Ariana Rowe, and I'm a fellow also with Condos. The title of our experiment is the impact of food dyes on oral bacteria.
First, we're going to talk about the dyes that we use in our food biomarkers.
But first, we should talk about FD&C. FD&C is a federal food, drug, and cosmetic act which is passed to allow 7 synthetic dyes to be put into our foods, drugs, and cosmetics. We will be testing 4 of those dyes today, which are red 40, red 3, yellow 5, and blue 1. However, the FDA is currently in the process of banning all FD&C dyes due to their synthetic nature. First is FD&C red 40, also known as myrrh red. The dye was originally developed in the 1970s. Red 40, similar to many other synthetic dyes, was originally derived from coal tar. However, present-day methods make use of petroleum. The compound is also classified as an azo dye, a synthetic organic compound. Red 40 is characterized by a bright red color and can be found in chips, sodas, candies, and other common products, including cosmetics and medications. Red 40 is commonly mixed with other dyes to achieve a desired specific color.
The next dye we're going to talk about is red 3 or erythrocene. It's responsible for putting foods a bright pink or red color and is commonly used in frostings, candies, and ingested drugs. The delating clause was passed by the FDA in 1960, and it states that a food dye or additive cannot be used if it is found to cause cancer in humans or animals. Red 3 has been found to cause cancer in male lab rats, and for that reason was revoked in January of 2025. The revoke process is scheduled to take 2 to 3 years.
The next dye is yellow 5 or tartine. This product is foods, drugs, cosmetics, chips, candies, drinks, mustards, even a bright synthetic yellow color. It is a byproduct of petroleum. It's composed of carbon, hydrogen, nitrogen, sodium, oxygen, and sulfur. Natural dyes are typically composed of carbon, nitrogen, and hydrogen. However, with the additional elements like sodium, oxygen, and sulfur, it gives an unnatural and synthetic dye. So the FDA is currently in the process of banning this dye because it has been seen to lead to cell replication, which can be very harmful to our bodies. The last dye that we're going to talk about is FD&C blue 1
bacteria that we tested on, which is oral bacteria.
So the oral microbiome is the second most diverse microbiome in the human body, second only to the gut. There are over 700 species of bacteria represented in 12 phylum. There are also various different niches throughout the mouth that have different types of bacteria. All of the bacteria we use came from the cheated saliva. Two of the most common types of bacteria found in the mouth are Fusobacterium nucleatum and various strains of Streptococcus bacteria, both of which were observed in our experiment. The oral microbiome plays a crucial role in maintaining homeostasis of the body. It also plays a role in protecting the oral cavity and preventing diseases from developing. Now we're going to talk a little bit about the experiment we conducted.
The question that we sought to answer was what was the impact of these dyes, red 40, yellow 5, and blue 1 have oral bacteria growth, and how does this affect overall health? The materials we used was McCormick food coloring, which we used yellow, red, and blue for. All of these dyes, however, did contain an added red 40 in them, so it was red 40 and yellow 5, and respectively with red 3 and blue as well. And we also used concentrated red 40 in our example food coloring, and the rest of the materials that we used for experiment are also. For our method, we first prepared 6 apple dishes. The first one was not treated with any solution, the second plate was topped with distilled water only, and the remaining 4 dishes were each topped with diluted solutions of their respective food dyes. Next, we obtained several pupil swabs and applied them to each of the plates. These plates were then placed in each air and checked almost daily for the duration of the experiment. Our hypothesis for the experiment is as follows. When exposed to different synthetic food dyes, the growth rates of bacteria will be impacted differently. We expect both of the control plates, that is the distilled water and no treatment plate, to show normal growth. Yellow 5 will show an increase in growth because it stimulates cell division. Red 3 and red 40 should both show similar results and that they will limit the growth of bacterial colonies. And blue 1 should show this limit on growth as well. Now we're on to the results from our experiment. Our first group, which was the no-treated group, received no water or dye of any sort, showed some growth, very minimal growth over the 6 days that we tested. You can see very small circular colonies that are very widely dispersed out on the plates or form, and this is what those colonies look like under the microscope on the final.
Our next control plate was the distilled water plate. As you can see, there's much more growth on this plate than the original control. We believe this is for hydration diseases, but we aren't really sure. We also believe that these are Streptococcus colonies, and they're circular in nature.
As seen in the series of images, the red 40 plate showed minimal growth over the duration of several days, and by day 6, there were 3 recognizable colonies on the plate, which we believe to be some strain of Streptococcus bacteria.
Our next plate was the red 40 plate. We saw similar results in this plate as we did in the red 40 plate, and that there was very few colonies, very little growth overall. On the final day, this is what it looks like under the microscope, and we believe that this is bacteria nucleatum.
Our next slide, yellow 5, you can see this growth over 2 days here. It definitely had the largest growth out of any of the dyes that we tested. This is what it looked like on the final day. You can see very strong colonies starting to form here, and this is what those colonies look like under the microscope. We believe to be informed by Streptococcus as well. And for our last slide, blue 1, it showed minimal growth up until day 4. And actually, if you look closely, you can see that blue 1, the bacteria growth is something different compared to all of our other examples. The bacteria started to grow on the edges rather than the middle. I believe this to be because according to the study used in blue 1, the dyes of diffusion and concentration rates can affect bacteria's growth. Certain areas can slowly and over become show only partial or no growth depending on the dye strength. So this could explain why the dye grew on the edges rather than the middle, because the concentration of our dye is high. Now our implications. Red 40 is often contaminated with trace amounts of benzene and formamidine. Both of these compounds are known to be carcinogens linked to leukemia and bladder cancer respectively. Furthermore, some studies have found correlation between red 40 and reduced attention spans in certain children, namely those with mutations in histamine degradation genes. Other studies have found red 40 to cause DNA damage and chronic inflammation in mice. The organs of mice exposed to red 40 were both larger than normal and dyed red. And lastly, as observed in our experiment, red 40 inhibited the growth of bacterial colonies. A similar experiment titled The Effects of Food Dyes on Gastrointestinal and Oral Normal Flora also reached a similar conclusion in cases of specific bacteria.
So next is our early implications. The study showed that it was effectively used as an antimicrobial, affecting various Streptococcus strains, and this is consistent with the results of our experiment in which we did not observe any Streptococcus strains on our red 40 plate. A second study showed that it significantly increased the risk of thyroid tumor cells and partially thyroid mice male rats, and this is the study that was ultimately growth is the authorization of design by the FDA. And a final study showed that in the gastrointestinal tracts of rats, even when they were given the approved dosage of Red 3 by the FDA, they saw decreased enzyme activity in kidney.
So for number 5, research in 2023 showed that this dye stimulates DNA and effectively damages certain DNA genes such as EMNT and HBC, which was responsible for regulating cell regulation in the body, particularly in bacteria. So every single experiment as this was played over right away, it had the strongest and most prominent growth out of the red dye plates. And that's even more significant considering that this dye was combined with red 40, which we observed to suppress the growth of bacteria. So immense bacterial replication can lead to a disruption of homeostasis in the mouth, which is in turn very bad for our oral health and just our general health. And so previous research has also found that this dye has caused itching, hives, and also hyperactivity in children. And then while there's no research done on humans, it is also to be predicted that tyrosine can be carcinogenic in our bodies due to the cell-regulated properties that it has. Blue light is often linked to hyperactivity in children and can also lead to possible brain and nerve damage. Long-term effects or high amounts can lead to harmful effects as well. In our experiment, we see that the bacteria only grow on the sides, not the center, possibly showing the effects of this dye. It is also a negatively charged dye, so it can mess with bacterial cell walls and it can cause damage and even kill the cell. Now on to some possible errors that we experienced during our experiment. Some of the dye got stuck on the sides of the graduated cylinders, so when we poured them into our agar dishes, it didn't all make it. And after 24 hours, there was still liquid remaining. So even when we swapped them with the bacteria, the swap soaked up some of the liquid from the agar. And then also when it came to storing it, we had to store it right side up rather than upside down and incubated.
So in conclusion, while there hasn't been much research done on humans in this field, synthetic dyes have many adverse impacts as shown by our research and our experiment. In our experiment, we showed that synthetic dyes can increase or decrease bacterial growth, which can disrupt homeostasis throughout the body. And our research showed that there's many adverse impacts that have already been proved in rats and also humans. Here are our sources, and thank you so much.
Alright, so good afternoon everyone. Thank you all for attending this presentation on microplastics. My name is Don. My name is Alan Malenko. I'm Salud. I'm Kia. So did you know that on average, the average human can ingest 5 grams of microplastics per week according to the University of New Mexico? Now, microplastics, we have to elaborate on what that is. So microplastics are small breakaway pieces of plastic from larger plastic debris such as plastic bags, bottles, clothing, and furniture. And microplastics can range in different types from fibers to particles and films. And they can also be found in different types of environments such as oceans, forests, farmland, and urban centers. And as their name suggests, microplastics are small, less than 5 millimeters in size, which is relatively the same size as a grain of rice. And there's also a subcategory of microplastics known as nanoplastics, which are less than 1 millimeter in size. And microplastics have become an emerging ecosystem threat due to the heavy human reliance on it. So humans rely heavily on microplastics in general, and this has led their footprint to grow at an alarmingly fast rate. Microplastics can be found in cosmetics, rain, trash, dust, and even table salt, according to Harvard Medicine. And despite this, microplastics are still a relatively new topic, so long-term human health and environmental effects are still unknown. Yet, despite this shallow knowledge, we should still be concerned because current research points to various harmful effects that microplastics can have on the environment and humans, such as biomagnification, soil degradation, and cell intrusion, as microplastics have been found to be able to bypass the cell membrane and thus harm the cell, as pathogens and toxins and bacteria can accumulate on plastics, and that's how cell intrusion is bad for the cell.
Just to add on to more of what Doc has said, we did an experiment conducting looking at the amount of microplastics in water sources. So we tested 4 water sources around here in college. We tested tap water, lake water, fountain water, and bottled water. So here, sorry. So again, we will analyze the amount of microplastics and nanoplastics in these water sources. So our hypothesis was that microplastics would be found in all water sources. We use a microscope, polarizing beakers, pipette, LED light, and petri dishes. So the purpose of this experiment was to show that microplastics can be found everywhere, that we should be mindful of the water that we drink and what we put into our bodies, and how microplastics can affect human health and the surrounding environment. So methodology. So first off, we prepared our samples. We poured each sample into petri dishes and made sure not to contaminate them. And then we cut out a square of pulverizing film and used that to put over each dish, and then use the LED blue light to look at the microplastics. So experimental errors before we get into our conclusion was that our method of examining was not actually accurate enough. Microplastics are really hard to look at due to their small size, and the polarizing and LED light was not an effective method of examining. So as stated, our experiment gave us insufficient result. One anomaly that we found was that we found black striations. So I did take a picture of the black striations, but it didn't come out clear and you could barely see them. You need to zoom in all the way. Anyways, alternative methods, due to budgeting, we weren't able to use the method that we wanted to, which was now red dye, which was meant to view microplastics. We could have bought a microplastics kit, but that's cheating. We could have used a stronger microscope, but we didn't have access to it. Anyways, problems.
So, as Adon touched on earlier, there are several problems, environmental problems surrounding microplastics. One of these is biomagnification. This is a process where as toxins like microplastics are absorbed and ingested by animals or organisms on the lower end of the trophic level and lower end of the food web, and as they're consumed by animals higher up on the trophic level, higher up on the food web, they are found in greater concentrations and greater quantities. And so this is a problem for several reasons. It can harm organisms in several ways. One, microplastics can carry toxins like pesticides, like heavy metals, like and other sorts of chemicals and pollutants that can directly cause harm to these organisms. Additionally, microplastics themselves can pose several risks. For instance, they can cause digestive issues. They can cause issues with the digestive tract to the point where the animals end up starving or even dying because of the buildup of microplastics in their bodies. And this can also then lead to humans because if, for instance, you're getting a fish from the ocean, and that fish has had ingested a large amount of microplastics and is then consumed by humans, these similar problems can be found. And so it leads us to our next slide. So the effects of humans that microplastics have. So this, like Adon mentioned earlier, this is still a fairly new field and the long-term effects have not been studied to a great extent. However, there is evidence that suggests they can have many problems like DNA damage, cell damage, gastrointestinal problems, and many other things as you see here. They can be very harmful and should be investigated more. And as you see here on these graphs, these are some sources of microplastics. The main one obviously is plastic debris. But there's several other sources that people may not be aware of. So like it can come from tire wear particles, from personal hygiene products, even from washing clothes. And then as for how microplastics get into our system, there are, once again, several different ways. From water and food ingestion, like we mentioned, but also through the air and through dust particles, additionally through cosmetics, they can be into your system. So it's important to be cognizant of all the ways that you could potentially be harming yourself by ingesting microplastics. And then on the far right, here are some different risks associated with microplastics. So food security is very important. You want to make sure that you're able to process food in a way that makes it safe for consumption and can try to limit the amount of microplastics found in your food and water.
So we researched 3 solutions. Our first was PETACE. PETACE is a plastic irrigating enzyme that targets polyethylene terephthalate, which are the most abundant plastics in the environment. And they were first researched by the researchers in Japan in 2016. And they can be useful for companies because plastics pollute the environment very heavily, and they can, the PETs can like break down microplastics for bacteria to use as fuel. Our next solution was for companies to switch to renewable resources. And this would help reduce the amount of pollution that plastics like pollute the environment. And our last solution was for consumer behavior to change. So this would help because if consumers opted for cleaner materials like cotton and wood, it would help companies like force their practices to change, and they would have to use cleaner materials.
So yeah, in conclusion, microplastics are an issue that can be found all throughout the environment. They not only impact humans, but also the animals that ingest it through things such as biomagnification, and they can also impact the environment through things such as soil degradation. And despite the already ubiquitous presence of microplastics in the environment and the limited knowledge on it, we as humans can still assume small consumer habits through consumerism to diminish the overall consequences it has.
And then I'd also like to thank our advisors, Megan, Kayla, and Juan, for a lot of their support and help throughout these 3 weeks. I'd also like to thank our fellow Cameron, who isn't here, but yeah, and I'd like to just give them a big round of applause. Watching our presentation on microplastics.
Any questions from the audience?
Is there a type of plastic that doesn't like emit microplastics?
So microplastics in general are made from bigger bigger pieces of plastic. So if plastic erodes, there are going to be small microplastics. So technically, to answer your question, yeah, all microplastics do create microplastics due to erosion.
What was your favorite part of the experiment?
Our favorite part of the experiment was probably like going down to the lake and getting the sample, the lake water sample. That was the best part.
Just went on a nice little walk there, buddy, just around campus, getting water. That was pretty fun.
It was a pretty nice day.
Take the point out, I didn't go on that walk. I was busy setting it up.
Yeah, I was wondering if you guys researched and looked into the cleanup process of
Microplastics. So right now, like the best way of cleaning up microplastics are through like filtering and like filtering and cleaner processes such as that. And another one that Sylvia mentioned was PETase. So in Japan, they researched this enzyme that breaks down some of the most common forms of plastics, so that bacteria can actually process them. So what we found was that those two would be the most reliable sources of cleaning out microplastics.
And I want to add on to that. So, I mean, obviously there's solutions, but the best thing to I guess, the best solution to microplastics is prevention. That's through like consumerism and buying things like biodegradable items and things such as cotton.
If humans ingest microplastic, where does it get stored?
So what we found was that it mainly gets stored in like your digestive tract or like in any other body. But for the most part, microplastics are generally something that hasn't been deeply researched yet. So the long-term effects aren't there, but the more that we are researching it, the more it's pointing to like these adverse effects of like to the human body.
Yeah, but they can be found like throughout your body. Like, they've been microplastics can be like in your brain, like you said, with digestive area, but it's still being completely fully studied. So.
Yeah, I have 2 questions. So technically, are you implying that we all have some kind of microplastic in our cells right now?
Yes.
But
Sorry, 2, do you think there's anyone in the world who would have microplastics within area?
Yes. I mean, I'm sure there's some like maybe like tribes in some sort of primitive areas that may not have been touched by plastic and waste at this point. It could be possible, but I mean, there's really no way to know.
So as you see both sides, but like, and plastic less than 5 milliliters, and then nano was less than 1 milliliter, and micro was less than 5 milliliters. So like, what's the most common to like plastic defined in your bottle?
So that part, nanoplastics would probably be the most common because like they can travel and they can be digested by your body pretty easily. I haven't found anything regarding like the most common types of plastics within like the human body since it's such a vague study. But from what I've seen, I think nanoplastics would probably be the one because like it digests probably the easiest.
And they're also like the hardest to detect in like in food or water or other sources. So those would be like those are probably the most common in whatever your food water is, and then therefore that'll lead to them being ingested more than normal microplastics.
But like if you just, I would like think that, but if you have like some amount like microplastics and nanoplastics in your body, like would you do more damage to you?
That's a good question. I'm not entirely sure because like we mentioned over and over again, they're still investigating the long-term effects, and so that's a difficult question to answer without sufficient research.
You mentioned using a type of red dye in order to find microplastics, ideally if you were able to. How does that help identify them or help you see them better?
So with the red dye, so let me first say this, microplastics have like different colors. With typically, if our experiment went as according, microplastics would give off like a red, blue, like different shades of color because like they're coming from different colors of plastics. By dyeing it with the red, it would essentially like kind of block out all other light and illuminate the microplastics.
Just real quick to add on to that, it doesn't necessarily, like we don't know for sure if that would have even helped. Like I said, we had pretty good microscopes available to us, but especially like in the bottled water, any microplastics found there are going to be extremely small to the point where we might not even have had enough magnification to pick them up. So there's no guarantee that would have helped. But obviously, that was the better overall method.
Since you guys have been talking about plastic for the last 3 weeks, is there anything that you guys noticed that was made out of plastic or like came across something plastic that you were kind of surprised?
Probably like the other day, I microwaved like some mac and cheese, and like it got me thinking. We've been doing this project on microplastics, and it like just made me think about like when plastic gets heated up, especially like in the microwave like that, like little particles are definitely like being broken off into the food, and so it made me like realize I probably should not like consume as much microwave mac and cheese.
So you talked about how like plastics can like enter your cell and like carry disease. Have there been any like recorded cases of microplastics causing disease like this or having someone?
So I haven't actually come across any cases such as those, but we found that microplastics can carry toxins, like very bad chemicals, and that has actually been found with some animals. Like their entire digestive tract like covered in plastics, not microplastics, but plastics in general, where like they die because of chemical pollution and plastic pollution.
I can add on to that. So I was also researching that, and I sort of found an article talking about like how if you have more microplastics, it can lead to like certain diseases such as dementia. But then again, it's also like we've been saying, it's relatively. So that's not concrete information. That's like sort of a weak correlation, but it might have a relation to like an increased chance for diseases.
All right, we'll have to stop there. Please thank group number 4.
These cells compensated for hidden for dormant for many years, and came back several years later, even after going through chemotherapy, hormone or other types of treatments. Breast cancer commonly spreads to the thalamus because of its rich blood supply and signals that attract cancer cells. Hormones like estrogen also play a very big role in this. A study done 2 years ago found that SDF1, a signaling molecule that was given out by thalamus, increased the chances of hormone receptors binding on to cancer cells. This process would increase the chances of metastasis, and many current treatments use hormone therapy to block off estrogen and slow the cancer down. All of these factors make breast cancer a complicated disease that requires ongoing research and treatment advances. Okay, so invasive species are characterized by many different things that set them apart from typical native species. Firstly, they can rapidly acquire nutrients as they tend to have a smaller leaf mass that allows for more efficient photosynthesis. Through this abundance and the complex root systems, they are more able to rapidly reproduce. And due to this growth, adaptations and mutations can happen more frequently as more and more DNA is mutated, allowing these species to survive in many different environments and possess immunity to various pathogens. More specifically, kudzu is an invasive plant common in North Carolina that was introduced in the late 1800s. It is a rapidly growing vine that covers large areas surrounding or covering the sun or covering plants from the sun. Its deep roots also help store energy and nutrients through large tubers that allow it to survive through droughts and other extreme conditions. Kudzu can also ruin habitats for wildlife and destroy telephone poles and buildings. Lastly, its roots or its leaflet releases nitric oxide into the soil, which can raise ozone and nitrogen levels in the environment. So although there is no true correlation between cancer cells and invasive species, the two do share similar behaviors that allow for uncontrolled growth and metastasis. These are three hallmarks of cancer that invasive species can somewhat bear. First, genome instability and mutations allow invasive species to develop specific immunities to new pathogens and adaptations to different environments. And although invasive species are inherently immortal and can resist cell death, their rapid spread and lack of natural predators lead to a growth that is hardly ever eradicated, or is hard to eradicate. And for the third one, invasive species perform their own version of angiogenesis as they take advantage of natural resources and restrict non-primitive organisms. They can also introduce harmful pathogens into the environment or alter soil conditions. And furthermore, kudzu and other invasive species can block sunlight and some other plants, effectively suppressing the growth of native organisms while supporting their growth. We chose to compare the characteristics of kudzu and cancer to explore the relationships they have in terms of behavior and hormonal properties. Both spread rapidly, disrupt their environments, and are difficult to control once they're established. Kudzu overtakes native ecosystems, much like how breast cancer overtakes healthy in the body. This comparison could inspire new strategies for detection and targeted treatment by revealing parallels in spreading, or like the way it spreads, and environmental interaction.
So yeah, kudzu and breast cancer, they're complete opposites, right? But what if we can use kudzu's mechanisms to suppress the spreading or growth of breast cancer? Okay, so kudzu is already applied in Asia as an anti-inflammatory and antioxidant agent. In the West, it's an alternative for hormone replacement therapy because it reduces the risk of getting breast cancer. Because kudzu has a phytogenic compound called that is an estrogen mimic that mimics the estrogen activity in the body. And in a study done this year in South Korea, found that this phytoestrogen property has an ability to induce cell death or apoptosis, especially for breast cancer. And they studied this and wanted to see if they were right. So these are the results. Unfortunately, it's tough, but you can see it. The isophagons, which is a type of phytoestrogen, kill cancer cells, not normal cells per day. For B, the isophagons actually shrink a tumor, as you can see here for this control image, all the way to its magnified. And for C, the isophagons stop the
depending on the type of isoflavor we use, can have multiple different effects. A solution that is actually simple, which is just use different types of isoflavors. Don't use the ones that you know will do opposite of what you want them to do. But that is actually really difficult, and recent studies today actually struggle with that same problem. They want new technology, new funding, and new clinical trials. Because if they can kill breast cancer in petri dishes, imagine how much progress they could make given the right therapies.
So for further future advancements, in a study by the University of Pittsburgh Cancer Institute, researchers explored the similarities between the invasive species English elm and cancerous glioma cells. In the study, both glioma cells and the tree seeds first grew in colonies, ultimately merging into a larger group with irregular borders. And in the study, the borders of the growth of the English elm and the glioma cells displayed an almost perfect fit, demonstrating similar growth patterns between the two. These similarities can potentially allow researchers to analyze and detect the early development of cancer cells. And as an honest, phytoestrogens only concern breast cancer, but ovarian and prostate cancer are also induced by estrogen. And we're only talking about estrogen here, since testosterone also does induce these cancers.
Now, this future cancer comes all down to prevention. Preventing the formation of cancer cells within the body is the best form of treatment for any cancer. So our pediatrician plays a large role in this, so it's important to have yearly checkups, screenings for cancer, and to perform multiple self-examinations for cancer multiple times a month. That's the presentation.
The Environmental Working Group, or EWG, is a nonprofit organization that advocates and does experiments for environmental health issues. We pay stereotypic clusters, picture to the right, and Southpath kids, picture to the left, because we hypothesize that they have many additives that would be dangerous to the germination of our seeds. These images depict why each candy is rated so low by the EWG. As one of their findings include that both candies are ultra-processed, contain many concerning amount of additives, quantities of sugar, and synthetic dyes.
In recent years, concerns about additives in our environment have increased, with modern research indicating the harmful nature of these with future implications for dangerous effects. These are now labeled as contaminants of emerging concern, or CECs. When plant organisms are exposed to CECs, they face numerous dysfunctions, diseases, and even premature death. This led us to look at some of our favorite candies with a long list of additives that are in With that in mind, we asked the question, what effect do artificial additives in candy have on the germination of seeds compared to sugar or water? We hypothesized that the candy solutions that had artificial additives would reduce germination and increase alcohol growth, meaning they had no mold or they had brown roots, compared to plain water or sugar. We suggested that those additives interfere with basic cell functions.
Okay, so before we get into our actual experiment, we want to explain what are artificial additives. So artificial additives are chemicals added to food to make it look, taste, feel better, and also to prolong its shelf life. Some common examples are artificial sweeteners like aspartame and artificial colors like red and yellow fire, and preservatives that keep our food from spoiling. These ingredients are commonly found in highly processed foods, just like the ones we used in our experiment. And even though they are approved for use in the U.S., there's growing concerns about how safe they really are, especially over constant consumption and exposure over time.
So you may be wondering why this matters. So germination is one of the simplest models for life and growth, and especially lentils, because as you can see in this image, they only need about a day to sprout. And so because of the anticipated presence of artificial additives in our environment, we wanted to see how the ones in our favorite and popular candies would affect plant development and that same germination. To create the concentration of our candies, we first started by crushing each solid into as small piece as we could, and we measured about 1 gram of each, so that would be our gum clusters, our Sour Patch Kids, and as well as granulated sugar. We added that to 10 milliliters of distilled water, and our control group of control. And then all 4 mixtures were left over the next 24 hours to fully dissolve and become more concentrated.
To germinate, lentils need oxygen, moisture, and water. In order to simulate the optimal conditions for our lentils, we took a 1/4 of paper towel and we dampened it with different solutions. The different solutions include distilled water, sugar water, Nerds Gummy Mustard solution, and Sour Patch Kids solution. While researching studies similar to that which we aim to carry out, we found that some artificial dyes reduce fruit and shoot development as well as germination rates in plants. The dyes that are concentrated in Nerds Gummy Mustards and Sour Patch Kids are yellow 5 and yellow 6, blue 1 and blue 2, and red 40. While little research exists around yellow 5 and 6, blue 1 and 2, red 40 is commonly known to be a harmful additive in our food. According to comparative studies carried out by other students similar to ourselves, red 40 can stunt plant growth and eventually lead to plant death. And the conditions that we created for our lentil seeds, normal growth was simulated by our picture.
Okay, now let's get into the exciting stuff we actually saw in the first experiment. So on day 1, all of our lentil seeds were placed on top of our classroom cabinet where they received intermittent light during the time in which that we had class. After 24 hours, we saw no seed growth across all of our test groups, including our water control, our sugar control, our merit extract, and sour patch extract. There were also no noticeable changes in the color or texture of our paper towels in the bags. At this point, the germination rate is of course solidified, which is 0%, which was expected because sprouting usually occurs after a couple of days.
By day 2, we started to see major differences across the test groups. In the water group, all 5 lentils successfully sprouted, with white roots measuring about 1 centimeter. There was visible condensation inside the bag, which suggested that there was healthy humidity levels. In contrast, the sugar water was some mold on the seeds. And the Sour Patch showed no change in appearance. There also wasn't any mold on the Sour Patch, suggesting that although growth was delayed, there was no immediate fungal activity. However, the Nerds Gummy Cluster group began to develop 2 mold spots on the paper towel, and the lentils also showed no signs of sprouting and remained the same color. Overall, by 48 hours, only the water group demonstrated successful and consistent seed germination. So by day 3, the water group showed strong and healthy growth with 1.5 centimeter sprouts, green tips, and no mold, meaning normal germination was happening. The sugar water group, however, had 1 yearly sprouting, and it also had mold starting to grow, so that suggested potentially hindering future growth for that one. And the Sour Patch Kid group, it still had no sprouts, and mold also started to appear in that one, suggesting how the additive maybe interfered with the germination. And most surprisingly, despite having the weakest sprouts and the most mold, the Nerds Gummy Cluster group had an 80% germination rate. By day 6, the water group had the healthiest of all growth. All 5 seeds were sprouted, and the roots were about 2 centimeters to 2.5 centimeters in length. They had green leaves, and no mold was seen either on the paper towel or the surface of the lentils themselves. The sugar water group, in contrast, only had 1 sprouted seed. It was very pale in color and was only about 0.2 centimeters in length. There was a little bit of mold on the surface of the lentils, but not really any on the paper towel itself. The Sour Patch Kid group had no germination period. There was also a lot of heavy mold seen on the paper towel and on top of the lentil surface. The Nerd Gummy Cluster group had 4 out of 5 seeds sprouted, and they were about 0.5 centimeters to 1.5 centimeters in length. They had white roots with browning on the ends, and they were surrounded by heavy dark black mold, and it was also seen on the surface of the lentils. While the water group supported the healthy germination, the other candy groups either delayed growth or stopped it completely, which was seen in our Sour Patch Kid group. And the presence of mold also showed that the sugar and the additives in these candies created a harmful environment for the seeds by feeding the microbes instead of feeding the plant itself.
So we have all this information, but now let's interpret it and figure out what it actually means. So we were able to conclude that additives in high sugar concentration subgrowth lentils. This suggests that these additives in highly processed sweeteners, if they enter our environment, they can inhibit growth of various plant species and microbes. The mold growth in our candy treatment suggests that high sugar and additives feed on fungal growth. This means that more mold equals more stress on seeds, which results in them to turn brown and to fail to develop. Sugar water had 1 sprout, which was surprising, by day 6, which tells us that too much water might interfere with water absorption, which is also called water pressure. And overall, our results partially support our hypothesis on the basis that UV solutions have more sunlight growth and abnormalities than the water group. However, the water group sunlight growth was similar to our other test groups, which we definitely were not expecting. We hypothesized this occurred due to the larger concentration of sugar than its other respective groups.
We were unable to record data on days 4 and 5 due to lack of accessibility to the lab of historical lentils. However, due to the predictable outcomes as seen in day 6, we can assume that there was a little bit of burden on those days. We were also unaware of the exact amount of light that the lentils were exposed to due to the different class schedules within the week. However, we know that they all have the same amount of light exposure because they were placed closely within the same area. Additionally, we were unable to get exact measurements whenever we were creating our solute because of the sticky nature of the candies. And for future endeavors, we recommend tracking long-term plant development and experimenting using natural candy alternatives such as fruit juices. Here are our references. Thank you.
To Governor School Week's Natural Science Cornerstone presentations. We are one of 4 rooms in which our natural science groups are going to be presenting their cornerstones. And today we have 4 groups that are excited to share what they have been working on for the past few weeks. So, in order to kind of set them up for success here, we do have JT in the back who will be recording all of the presentations, and you will have access to those after all this. At some point in the near future, you'll have access to the video recordings. And we also ask that you silence all cell phones if you would please. And if you have your cell phone out, it needs to be face down for whatever reason, do that as well. And then if at any point you do need to leave the room, we just ask that you do so quietly and re-enter quietly. And just about does it. So we have 4 groups who are prepared to share with you what they've been working on, working very hard on for the past 3 weeks. Each group is going to have approximately 10 minutes, okay, to present their basically what they did. And then there's going to be a few minutes after this if there are any questions. Okay? And then there's just going to be a brief transition period between groups. All right. Well, I feel like it is time for us to get started. Okay? Please welcome our first group to join us.
Hi everybody, this is Toxic Tides, investigating pollution in water systems. I'm Mason. I'm Izzy. I'm Shreya. So to take you through this, first we're going to introduce you to our idea, research behind it, and our hypothesis, and then we're going to then go to our experiment and the data analysis and collection data. And then we're going to go through some evaluations, comparisons, reflections, and a surprise at the end.
So to start off our research, we had to pick 3 chemicals for our brine shrimp to see how they would survive. So for the 3 pollutants we chose was bone meal, flour fertilizer, and phosphate detergent. This was kind of chosen based on convenience. It was already supplied by our instructors. And so we did some research on the pollutants to understand the chemical composition of it. So for the bone meal, there is 6% nitrogen and there is 8% phosphate. And we're specifically going to look at nitrogen and phosphate because that affected the brine shrimp the most. Then we looked at flour fertilizer, which has 10% nitrogen, and here's the breakdown of nitrogen. And then there's 10% phosphate. Then finally, for phosphate detergents, the chemicals were not on the back of the bag, so we had to reach out to the company to understand what the chemical composition of it was. And they responded to us, so these are the details we got. And it is high in they're mini versions of shrimp. And basically we had eggs that were already supplied by our instructors and we had to grow them. So we had to do some research on how long it would take to grow the brine shrimp, as well as what kind of waters they thrive in. So we found out that brine shrimps take 24 to 48 hours to hatch. And then to simulate the ocean water, we found that 35 grams of dissolved salt in each liter of water creates seawater. So we did 500 milliliters of water, so we used 17.5 grams of salt and mixed that into the water to simulate the oceanic water.
Okay, per request of our instructors, they wanted to see the brine shrimp, and I think that's a great idea. So here's a video of them swimming around. So they're really anyway. And then these are also some pictures of them. As you can see, they're really small. You use a microscope to see them. And imagine having to count all of these.
Okay, so our hypothesis was, if brine shrimp are exposed to these common pollutants, then their survival rate will decrease in all samples. Specifically, we predict the brine shrimp exposed to bone meal will have the highest survival rate.
So for our experiment, we started off by growing the brine shrimp, and as I said earlier, we simulated the ocean water, and then we put some brine shrimp eggs. Then we monitored how long it would take to grow, and it grew in about 24 to 40 hours. Then we measured out chemicals. We chose to do 0.1 and 0.4 to see if 0.1 would allow the brine shrimp to survive, and if 0.4 would cause it to die out. And then we mixed the chemicals into the brine shrimp, but we did them in petri dishes so that the chemicals would be isolated. Then over a period of 5 days, we counted the brine shrimp and saw how many survived throughout the days. And we also counted the eggs to see if any of the eggs hatched. And then for the data, you can see here that there's all of them are alive in the beginning, but then when you get to day 3, most of them have died out except for bone meal 0.1 and phosphate 0.1. But then you also see the increase in bone meal 0.1 from day 1 to day 3 because some of the eggs hatch. And then when you get to day 5, everything has died out except bone meal, and some of the previous ones from day 3 have also died out.
Okay, so at first, all our groups were pretty healthy. We had enough for brine shrimp each. By day 3, almost all of them had died except for 0.1 bone meal, which still had 40 alive. By day 5, bone meal had dropped to 29. And then we found out that if you add higher amounts of these chemicals, then even the ones that don't kill them kill them. So for the chemicals, bone meal was much less harmful than fertilizer and phosphate detergent. Fertilizers and phosphate detergents cause an increase in promotion of algal growth, which causes a decrease in oxygen. And then bone meal releases its nutrients more slowly, so it reduces immediate harm.
Okay, so to think about how this goes like large scale global, we have to think about how this can represent those fertilizers from agriculture getting into our water sources. So from runoff, like the aquatic organisms that we know love, like fish, they can't live in super heavy polluted waters. So we have a loss of biodiversity, and we can even have things like biomagnification, which can then affect us. So to reflect on our time, some successes we had was that our bone meal result matched our hypothesis. We had the conditions kept very stable the whole time, and then we learned how to use our equipment properly. Challenges, however, were estimating how many brine shrimp we had because they were so small, and then release amounts, keeping them approximate, and then maintaining a livable environment for them. Errors, having old like shrimp eggs, these weren't the first ones to use, and then miscounting them since they're super, super small, and then those companies not being totally informed by the ingredients.
Okay, so these are the graphs that we made. As you can see, day 1, we had a lot of alive ones and still eggs. Like I said before, bone meal was the least harmful. By day 3, we had a little bit of phosphate, but mostly just 0.1 bone meal. And then by day 5, it was just the bone meal. It got kind of hard to tell which were eggs with like the phosphate and the fertilizers, so we didn't end up counting the eggs in the later days.
Okay, now y'all are gonna be stuck with me for a little bit, and I get to talk about our little surprise that we had. So yeah, on day 5, we found something unknown in one of our dishes that were closed. We weren't sure what it was. Our instructors first went, "Who lives in Maine?" Because they thought it was a flea. After really looking at it, we just noticed there was antennas, and that means it's not a flea. It ended up being springtails. So we did some research about springtails. They live in soils, like moist soils, underneath litter and stuff. They eat dead, decaying matter. And they are attracted to like moist areas, but not salt water. They don't live in salt water. Like you know how spiders float on top of the water, they do the same thing as that. They do not live in water. So what does this mean? Why did it happen? We had some guesses on what could have possibly been like the cause of this. They were either already present in the materials we used, like fertilizers and stuff, and so when we introduced those into the dishes, then maybe the eggs hatched and they spread. But this didn't really make sense because they were only in 5 to 6 dishes, and when we looked at them under the microscopes, like this one here, they were dead. Or they came in as airborne contaminants before the dishes were sealed. I quickly eliminated this because the controls were not sealed. So why was there none of the controls? And why was there none of the food? Okay, so then, even though they were dead, they were somehow reproducing. We did suspect that they were asexual, so maybe this could happen, but why would they want to reproduce in an environment they're not used to and they don't survive in? This is a video of all of them. It was kind of scary. There was a lot of them. And after many days, they were spreading to the other dishes, and they're even there so now. So what did we do about it? We asked our instructors. They were little to no help. They weren't. They don't know that much about bugs. So we talked to somebody smarter than us and smarter than our instructors in this area. Okay, and so we went to the NC State website and we looked for entomologists, and I emailed one. And this is me being super official, which, if you know me, not like me at all. And he reached out and he got back to us in a day. So yeah, that was really cool. Basically, he confirmed the species, which is really important to do further research on them. And then he also gave us like maybe an idea of what it could be. He thought it was the water, which I don't even think about for one. But I quickly ruled this out because it was not in our control and we used the same water for control. Now this is still an ongoing experiment, and we are still like seeing them pop up, and they're still reproducing. And I'm emailing the guys still. I'm waiting for him to respond again, and we're still learning more. Thank you.
Do you think that the organisms that you found affected the number of brine shrimp as well? Did they have any effect on the brine shrimp?
No, because we didn't really care because it was the end of our experiment. They showed up day 5. That was the end of our experiment. I don't think they had any effect on it personally. The ones that are still living to this day are a lot larger than our control, so maybe they're eating them, but not
Where did you get the water from? Was that Meredith water?
Yeah. Oh my God. applicable in the real world, our question and our hypothesis, the materials that we use, our experimental procedure, the analysis with the results of our procedure, the analysis of our results, and our conclusion. So what exactly is seed germination? Seed germination is a process in which a seed transforms into a growing plant. It requires a combination of warmth, water, oxygen, and NPK, which is the most important nutrient. It stands for nitrogen, phosphorus, and potassium. And with all this together, it's how they activate their seed's initial root and shoot growth. We chose to focus specifically on root growth and not shoot growth, as root growth determines the ability of a plant to take up nutrients and water. A stronger root system can perform these tasks more efficiently with a greater ability and overall strong and creates a stronger plant. Whereas a weaker root system cannot perform the task as well and it has a lesser ability and overall weaker plant is formed. So what exactly is nitrate? Nitrate is a natural compound that is composed of oxygen and nitrogen atoms. In relation to plant growth, they're primarily used after germination, but small amounts of nitrate can promote seed germination. Phosphate is a compound made up of one phosphorus atom that is bonded to 4 oxygen atoms. Phosphate enhances the nodulation of seeds during germination. Nodulation, in relation to plant growth, is the process of forming small root nodules. This leads to improved nitrogen fixation and overall stronger plant growth. So why is our project relevant? 55% of all American households do garden in some capacity. This is equivalent to 71.5 million households or 185 million people. Tap water is typically the most common water used on plants. I know in my personal experience, I currently still use tap water for my plants, and I have used tap water to germinate my flowers. And a germination rate affects crop yields. A greater germination rate means it has a greater yield, but a poor germination rate has a poor yield overall in crops.
So the question we're seeking to answer is what type of water, considering factors of nitrate and phosphate, is the most effective at germinating plants? Our hypothesis was that if we use lake water, which is typically rich in nitrate and phosphate, then it will have the greatest rate of germination compared to the less nutrient-rich water samples. So the materials we used were 6 glass jars of lids, which we kept the water in, 6 plastic bags, nitrate and phosphate test tablets, water test strips, lentil seeds, a microscope slides, of course, grow light, paper towels, pipettes, and various beakers. So step 1, we had to gather the water from the sources. We got water from the lake on campus, the fountain in the quad, the water in the cafeteria, a puddle on the sidewalk from the library, tap water from the dorm, and the still water. Each of these samples was kept in a glass jar inside of the drawer. Step 2, bacteria testing. To test for bacteria and microplastics in dirt, we put 2 drops of each water sample on a microscope slide and observed it through 4 times magnification. Aside from dirt, we did not find much relevant data in this part. For nitrate and phosphate testing, we use nitrate and phosphate test tablets and strips to test how many parts per million each sample had. For nitrate, it was 5 milliliters of water to 1 tablet. For phosphate, it was 10 milliliters of water to 1 tablet.
On step 4, we began seed germination. We did this by applying the water samples to a paper towel with lentil seeds and placing it into a plastic bag. Then the bags were all placed under a grow light. We then observed and charted their average root growth over the course of 6 days. This graph details the nitrate levels of each sample in part per million. Cafeteria water had 6 parts per million. Lake had 5. Fountain had 3. Tap had 5. Distilled had 2, which was the lowest, and standing had the highest at 7. Now on this graph, we are showing phosphate levels in each water sample part per million. In this one, cafeteria water was 2, lake was 4, fountain was 2, tap was 4, distilled now having the highest at 5, and standing was now lower at 2. Our final graph highlights the average root length growth over time for our lentil seeds. We
highest growth rate overall and the most consistency, and standing having the lowest overall without water.
Alright, so we were told to include more pictures of our seeds inside the slides. So we did that, but we also wanted to represent what we actually did in our process and how we measured the root growth, as well as I also wanted to bring up on the right side, maybe hard to see, but those roots right there are the standing water root growth, and it's kind of hard to see, but they had the thinnest roots out of all the other water sources, as well as you can tell them prematurely coming out of their shell. So that kind of highlights how the standing water was not very healthy. So don't drink standing water. Okay. And so onto our analysis, we found no major correlation between the levels of nitrate and phosphate and the rate of growth in the germination process as rooters. However, some of the fastest growth rates occurred with balanced and moderate levels of nitrate and phosphate. So this suggests that a seed will perform the best in the germination process with balanced levels of nitrate and phosphate. However, there was an outlier that did not match our analysis, and this was distilled water. It produced the fastest rate of growth, but with unbalanced levels. It had the lowest level of nitrate and the highest levels of phosphate from all other water sources that we collected. And this idea of unbalanced also was represented in standing water, but the exact opposite. So it had the highest level of nitrate and the lowest level of phosphate out of all water sources. This also produced the opposite results, resulting in the lowest rate of growth compared to all the other water sources. Still, we went with the majority results of the water sources, which was that they thrive in balanced levels of nitrate and phosphate. So, onto our conclusion, distilled water did lead to the greatest rate of growth, and this refuted our hypothesis that the lake water would yield the greatest rate of growth in the germination process and overall root growth. So, some potential reasons for this is that distilled is very pure and has no chemicals that could be potentially harmful for the seed, as well as it stripped away the essential materials that was needed in the germination process. It also has no microparticles that can obstruct the germination process, like all other water sources that we collected. Alright. And on to the potential experimental errors. So the first one that we had is inadequate bacteria testing. While we did test bacteria, we tested one sample per water source, and the issue is with this, while we did not find anything, the main issue with this is that just one sample does not represent the entire composition of the entire water source. So we needed to combat this misrepresentation the next time, we need to do multiple samples per water source so we can accurately represent the actual water source. The next one is the seed's potential exposure to inconsistent temperatures. We did have our seeds the entire time under the growth light inside the natural science building in our classroom, but depending on we did not record the temperature, so that might have been a mistake there, but depending on our professor's preference that day, she might have turned up the temperature or lowered the temperature, so but we don't think that had any effect on the seeds as they still were going through the germination process as normal and had condensation inside the bag. So the next one is our root measurement method. How we measured them was with bendable rulers, and just by human error, we definitely could have missed a centimeter or two. And here are sources. And then that's it. So thank you for joining us.
Why did you use lentil seed?
That's what we had available, and lentil seeds are one of the fastest growing plants, so they get nice, fast results over many days. We chose lentils. Yeah, we also, our first project inside of natural science was with Kayla, and that was the first thing we did was grow lentil seeds, so just incorporate that idea. Good answer. The worst was the standing water. On the
the other slide, it showed that the seed, actually all the roots were very, very thin, as well as they were coming out of their shell prematurely. So like I said, it's definitely not going to drink water from the ground, specifically outside the library. The biggest obstacle for me, I know, was measuring the actual roots. We did switch to a bendable ruler because a lot of the roots started curving, so that was a bit there. Another big obstacle we had was we were originally testing the pH in bacteria, but the pHs were all very similar within 6.8 to 7.2. And in bacteria, like you said, we even went to 100 times magnification, we could not see any bacteria. Any other questions? All right, thank you.
Our research was on the survival of E. coli and natural and synthetic antibacterials. So to start, let's give some background about our research. One of the biggest problems in American healthcare today is the development of antibacterial resistance. As resistance to synthetic antibacterials increases, it becomes necessary to find alternative options. According to the CDC, in 2019, over 5 million deaths worldwide were associated with antibacterial resistance, and of those, 35,000 occur in the United States. 2.8 million antimicrobial resistant infections occur in the United States every year, and over $4.6 billion are spent by the United States treating infections caused by these resistant bacteria. So these statistics just serve to show the severity of the issue. As you can see, it's extremely necessary to find an alternative option. So for this experiment, the alternative that we decided to focus on were natural antibacterials, specifically tea tree oil.
Okay, so throughout our experiment, we had one major question that we focused on. How will E. coli survivorship rates compare when exposed to synthetic antibacterials, max, versus natural antibacterials? We also had two hypotheses that we focused on throughout our experiment. The first being, the petri dishes treated with our synthetic antibacterial would have fewer surviving bacterial colonies compared to our natural antibacterial. Our second one was applying a higher concentration of our antibacterials, both synthetic and natural, would lead to fewer surviving bacterial colonies.
And this is just a quick overview of the procedure that we used. The first thing we did was we treated our petri dishes with two different antibacterials. The first one being tea tree oil, a naturally derived one. The second being Bactine Max, a topical over-the-counter antibacterial. And the third group was the control group, and that was treated with deionized water. After that, we inoculated the plates with E. coli and then incubated them. And while they were incubating, we took observations at the 24 and 48 hour marks and tried to count the number of bacteria observed.
So on day one of our procedure, we prepared all 6 of our solutions. First, we had to prepare the standard topical dosage of our tea tree oil, which consisted of 5% tea tree oil and 95% water. This was used as a full strength concentration of our natural antibacterial. This was further diluted to create our 75 and then 50% concentration.
Alright, and after analyzing the images for bacterial growth, what we noticed was that the control exhibited a good bit of E. coli growth, as you can see in that big spot right there. But the tea tree oil at every concentration and the Bacti max at every concentration did not exhibit any bacterial growth compared to the control. And based on these results, we showed that this kind of shows that both are effective options, the natural and synthetic, work decently well, and the concentration didn't really have an effect in our experiment with the setup we were using, and the natural and synthetic antibiotics both had similar effects in our experimental setup.
Of course, in every experiment there's going to be errors, so these are the four that we thought maybe had the most impact on our results. To start, the tea tree oil and the water made a heterogeneous mixture, which basically means they mixed in a way that allowed them to be separated. What was happening was the water was at the bottom of our petri dish, and then the tea tree oil was just kind of floating on top of it. So we weren't really sure if all the tea tree oil absorbed the nutrient agar, which might have altered the bacteria growth. Additionally, there was a high risk of cross-contamination just through unclean gloves, unclean glassware, and there was also one situation where the graduate cylinder hit the inside of a petri dish, which could have introduced some unknown pathogens into our experiment.
Another issue that could have affected our results was having excess liquids in our petri dishes. We added 3 milliliters to all of our dishes, waited 24 hours for them to hopefully absorb. They did not, so we waited another 24 hours hoping that they would absorb more. They still did not. This led to issues with our incubation, considering we could not invert our plates to reduce condensation. Because of this, lots of condensation formed on the top of our plates, which could have added extra water into our bacteria, which could have stunted our bacterial growth. Also, we are only humans, so human error is to be expected. This could have shown up in the form of inaccurate measurements, issues with our equipment, or just issues in our general procedure.
And from the errors that we observed in our experiment, we noticed some things that we could do in future experiments and that we can take from what we did in this one. The first thing we would do is try to ensure even mixing of the tea tree oil and water. So that could be by means of either keeping it under constant stirring while we're working with it, or using some kind of emulsifier to keep it a homogeneous mixture. The next thing would be to just ensure that all of our glassware and things we use are clean and sterile before using it to work with the bacteria. And then finally, making sure that we add a smaller amount of the treatments so that we are able to invert our petri dishes and incubate them in the standard way.
Okay, now on to applications. There are many future applications for our results in our experiment, but we will focus on just 2 today. The first one is antibacterial alternatives. Based on our results with these 2 specific antibacterials showing similar effectiveness in suppressing growth for patients with allergies to synthetic treatments or patients who may just prefer holistic or natural alternatives, this gives hope to future integration into standard healthcare practices.
And as previously mentioned, a huge issue today is the fact that bacteria is becoming resistant to many synthetic drugs, so it's really hard for infections to be treated. This is because most of these drugs will only target one specific process in the bacteria. For example, for penicillin, it will only inhibit the cell wall synthesis of the bacteria, so it's really easy for future generations to mutate and evolve to prevent damage that the penicillin does, making the drug completely ineffective. However, the 2 treatments that we use, our synthetic antibacterial and our natural tea tree oil, both target multiple different processes in the bacteria, not just one. They damage the cell membrane as penicillin does, while also denatures enzymes and other proteins, interferes with the metabolism and homeostasis of the bacteria, and even leaks out cell contents. It's almost impossible for bacteria to genetically adapt in so many different ways all at once, so it's much harder for the bacteria to develop resistance to both of the treatment methods that we tested, as opposed to many commonly used synthetic methods.
Finally, we have some acknowledgements. Thank you to our extremely educated and knowledgeable advisor, as well as Cameron for guidance on our procedure, advice on what to do, and helping redirect our experiment when things could have went wrong. Our error analysis would have been much longer without you. Thank you to Kayla and Juan, our other instructors, for continual support, advice in the early days of our experiment, and providing materials. And also thank you to our wonderful front desk lady, Rebecca Jensen, for being pleasantly disappointed at Carolina Biological for losing our first order of E. coli. That order is still in the mail. We don't know where it is.
Oh, that's fire. Thank you all for coming.
Why is E. coli used in bacterial experiments?
I can answer this. So we wanted to pick a bacteria first that was cheap and we couldn't find this. So we found a site that would let us do that. But we also wanted to use a certain kind of bacteria that you would find in a cut or something that you would use your vaccine or tea tree oil on. So we could have gotten bacteria that was like cheese bacteria or something, but you're not going to put tea tree oil or vaccine on that kind of bacteria. So we got E. coli because it is a bacteria that is typically found in injuries that you would treat with our antibacterials that we use.
Big sister question to that, why tea tree oil?
I think we use tea tree oil for similar reasons, because we already have the vaccine and we also already have the E. coli. We were like,